Hey guys, my name is Didi Jimenez, and today we're going to go over the structure and function of a single synapse. What is a synapse, you may ask? A synapse is the area where two neurons connect and communicate, aka talk to each other. This is done through a combination of electrical and chemical signals. Luckily for us, the picture labels a synapse to help us get a better visual. The synapse consists of three major parts: the presynaptic terminal, the postsynaptic terminal, and the synaptic cleft. The presynaptic terminal is located at the end of an axon. This is where the electrical signal is converted into the chemical signal. The postsynaptic terminal is less than 50 nanometers away from the presynaptic terminal and consists of specialized receptors. The synaptic cleft is the space between both the presynaptic and postsynaptic terminals. Now, let's take a look at the function. To put it easy, the synapse functions in transmitting impulses. This area also allows for integration, meaning an impulse can travel down a neuron and reach a synapse that has different synaptic neurons that go in different directions, resulting in the impulses being dispersed. Interestingly enough, it can work in both directions, in reverse. Now, those were just the basics. Let's take a look at synaptic transmission, which is the mechanism for communication among nerve cells with each other, or muscles and glands. Transmission begins at the presynaptic neuron after a firing action happens and goes to the postsynaptic neuron. This mechanism can be seen in two forms: electrical transmission and chemical transmission. In chemical transmission, communication happens using chemical messengers or neurotransmitters, which carry information from the presynaptic neuron to the postsynaptic. These messengers are found in vesicles that aid in transporting them to the terminal. This transmission is the most common, yet the most complicated, but we'll go in depth later. Electrical transmission, on the other hand, occurs through a direct physical connection and therefore is faster than chemical signaling. For this mechanism, a gap junction channel allows current ions to flow directly from one cell to another. The downside to this process is it can turn an excitatory signal in one neuron into an inhibitory signal in another. It lacks versatility, flexibility, and capacity for signal modulation that we see in chemical synapse. The benefits: it's faster. It allows for synchronized activity of group cells. It can carry currents in both directions, so that depolarization of a postsynaptic neuron will lead to depolarization of a presynaptic neuron, which kind of breaks the definition of pre and postsynaptic. Easy, right? Let's now bring our focus to the complicated transmission, aka chemical transmission. The easiest way to take this on is by looking at it in steps. We have synthesis, which happens in the cell body. The neurotransmitter is synthesized and transported down the microtubules of the axon to the presynaptic terminal, or it can be directly synthesized in the presynaptic terminal from recycled neurotransmitters, which are then stored in presynaptic vesicles until they're released. And then part 2, the release. The neurotransmitter is released from the presynaptic neuron, where the synaptic cleft receives it, leading to step 3, receptor activation, where the neurotransmitters bind to postsynaptic receptors and produce a reaction in the receiving neuron. And lastly, we have our signal termination, which must occur by some mechanism. Typically, it's by elimination of the extra neurotransmitter from the synaptic cleft. Let's now go over the steps one by one to get a deeper understanding. Depending on the type of neurotransmitters, synthesis occurs differently. A neurotransmitter can be a small molecule chemical, like dopamine and serotonin, or they can be small neuropeptides, such as enkephalin. Furthermore, neuropeptides are synthesized using protein synthesis and translation pathways. Then, they are packaged into vesicles with protease. After this comes their transportation through the axon using microtubular proteins like kinase. They can then be released once they arrive to the presynaptic terminal. Small molecule neurotransmitters are synthesized within the cell body, where they transport down the axon in small clear core vesicles once they arrive at the presynaptic terminal. Enzymes aid in modifying the small molecule neurotransmitter, where they can be released from the vesicle into the cleft. The next step is release. At this point, the neurotransmitter can be released into the cleft. Along the vesicle and presynaptic membranes, you can find snare proteins, which are essential in binding vesicles to the membrane and aid to release their contents. The membrane will depolarize as the action potential goes down, and presynaptic neuron. After the action potential arrives at the presynaptic terminal, there will be a rapid influx of calcium into the presynaptic terminal because the voltage-dependent calcium channels are open due to depolarization. This increase of calcium causes snare proteins to activate and change conformation, leading to the fusion of vesicles to the membrane and release of the contents. The synaptic cleft will then receive the neurotransmitters. With endocytosis, the vesicle membrane will be released. After the release comes the receptor activation. After the binding of the neurotransmitter to the postsynaptic neuron, you can get two types of activated receptors: ligand-gated ion channels and G-protein receptors. Ligand-gated ion channels are when the neurotransmitter binds with this receptor, you get direct opening and closing of the attached ion channel. To put it easy, the neurotransmitter acts directly on the target ion channel. This receptor type is fast. It produces responses in milliseconds and terminates quickly. These receptors can be either excitatory or inhibitory, depending on the neurotransmitter bonded. G-protein receptors activate a signaling cascade involving secondary messengers in order to produce a response. The most common secondary messengers are cyclic adenosine monophosphate, inositol triphosphate, and diacyclic glycerol. Once binding to the receptor, the neurotransmitter activates the G-protein, which binds to the guanosine triphosphate and is activated. After this occurs, the secondary messenger cascade is activated, and eventually phosphorylation of ion channels occurs. Because of all the steps involved, this is considered a slow mechanism with longer lasting effects, from seconds to even minutes. The last step is signal termination, which involves the clearing of the neurotransmitter in order to inactivate signaling. This is done in three ways: reuptake, enzymatic destruction, and diffusion. In reuptake, only small molecule chemical transmitters can be taken back up, except neuropeptides. Those are eliminated through degradation. There's two types of reuptake: presynaptic reuptake, which is the neuron will use different methods, endocytosis or specific transporters, to get rid of neurotransmitters from the synapse. The advantage of this is that neurotransmitters can be recycled. It prevents resynthesis of the neurotransmitter every cycle of release. And then the other type of reuptake is a glial cell, which will have to be utilized for reuptake in some cases, like with glutamate. It is toxic to a cell and therefore cannot be exposed to it. It is held inside of the neuron as glutamine. In this case, a glial cell is used to transport it using a special transporter, where it is converted into glutamine via glutaminase and then returned to the neuron for recycling. The second type of signal termination is enzymatic destruction, which is a direct destruction of the neurotransmitter within the cleft or in the presynaptic terminal by the use of specific enzymes. Two major ones are involved in the process: monoamine oxidase, which is responsible for oxidation or inactivation of monoamines. Inactivation is done by removing the amine group from oxygen. They are split into two: MAOA, which breaks down serotonin, melatonin, norepinephrine, and epinephrine, and MAOB, phenethylamine and benzylamine, based on substrates. Both break down dopamine, tyramine, and tryptamine. And then our other enzyme is catechol-O-methyltransferase, which degrades catecholamines like dopamine, epinephrine, norepinephrine, as well as most substances with catechol structures. Both of these enzymes are used in therapeutic medications. An elimination of them results in the neurotransmitter staying in the synapse more time, benefiting an elimination of symptoms of many disease processes. Our last signal termination is diffusion, which is the simplest form of termination. Neurotransmitter can simply diffuse out of the synaptic cleft and away from receptors and into nearby blood vessels. This results in a decrease of concentration in the neurotransmitter in the synapse and will gradually reduce the effect it has on the postsynaptic neuron. Now, why is all this important? As we know, synapse is a fundamental unit of neural communication. In this case, we're going to have myasthenia gravis, which is pathogenesis involves diminished communication between the neuron and muscle at the neuromuscular junction. This is because there is a blockage or destruction of acetylcholine receptors at the neuromuscular junction because of antibodies. This prevents acetylcholine from binding and depolarizing the muscle, which doesn't allow for contraction, aka blockage of receptor activation. In this picture, we have a 43-year-old patient that presented for a laparoscopic cholecystectomy and an intraoperative cholangiogram.
Why is this important?
This patient had no past
In the
medical
article
history.
I used, I found
They were in overall good health, not a
diseases.
smoker, not a drinker.
Myasthenia gravis, which is an autoimmune disease that causes muscle weakness.
Difficulty breathing.
Lambert-Eaton syndrome, which is an autoimmune disease that causes dysfunction
and was unable to raise their head and sustain
at the
a hand grip.
neuromuscular junction.
Different actions were taken to better the patient, but
and
after 10 minutes there was no improvement.
Bacteria-produced
so the patient was
toxins are the causative agents of both of these diseases.
Using propofol 100 milligrams.
As we know, the synapse is a fundamental unit of neurocommunication. So, in myasthenia gravis, pathogenesis involves diminished communication between the neuron and muscle at the neuromuscular junction. This is because there is a blockage of destruction of acetylcholine receptors at the neuromuscular junction because of antibodies. This prevents acetylcholine from binding and depolarizing the muscle, which doesn't allow for contraction,
After all these results, the patient was thought to possibly have an underlying neuromuscular disorder.
In Lambert-Eaton syndrome,
the patient was then released and then readmitted.
Antibodies are directed against the calcium channels on the presynaptic neuron, preventing calcium influx from happening. Because of this fusion of vesicles with the presynaptic membrane and the release of neurotransmitters into the synapse,
the patient
is also
was
prevented,
aka
prevent
muscles,
neurotransmitter release.
Extensors, and fatigable proximal
Botulism and tetanus. This toxin acts as a protease that cleaves the snare protein.
Was discussed with
by inhibiting vesicular fusion, this prevents the release of neurotransmitters at the junction.
A CT scan, the one you see
botulism
in the picture, showed an
Clostridium
botulinum
with produces the botulinum toxin that prohibits the release of acetylcholine, the stimulatory neurotransmitter, which inhibits stimulatory effects which prohibits muscle contraction and causes flaccid paralysis. Tetanus. Clostridium tetani produces the tetanus toxin that prohibits the release of GABA and glycine, inhibitory neurotransmitters. For this disease, their release is inhibited in the spinal cord, specifically Renshaw cells. Symptoms include spastic paralysis, locked jaw, and opistenosis, which resembles an upper motor neuron lesion.
Myasthenia gravis.
These are just examples of how
autoantibodies
they affect
nicotinic AChR at the neuromuscular junction, which reduces the number of functioning receptors, either by competitive block, increased degradation, or receptors, or by complement-mediated lysis of receptors. So I would say that the importance of all this is that this is the blockage phase of what we just went over, considering that it blocked receptor activation, which was our third step for chemical transmission. Well, after all that information, we have now reached the end of our presentation. There you have our resources in case you want to look more into it and get a deeper understanding. Thank you guys. See you on the next one.